Welcome to today's live broadcast, Comprehensive Characterization of a Monoclonal Antibody in a Single Analysis. I'm Stephanie Warth from the Division of Medical Sciences at the University of Victoria, and I'll be moderating your webinar today. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by SCIEX Separations, a part of AB SCIEX. Capillary electrophoresis and liquid chromatography are among the most powerful analytical tools available to scientists today. AB SCIEX is expanding the power of these technologies by combining Beckman-Coulter capillary electrophoresis and exigent liquid chromatography to create the business unit SCIEX Separations. Particular capillary electrophoresis instruments and all liquid chromatography instruments are designed for connectivity to mass spectrometers from AB SCIEX and other leading manufacturers. For more information, please visit www.sciex.com. Before we get to today's presentation, I have a few announcements. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the question and answer box, which is found by clicking the green Q&A button in the lower left of the presentation window, and we're going to try and answer as many questions today as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button in the lower left. Our presenter today is Brian Fonslow. He received his PhD in analytical chemistry from the University of Minnesota, where he worked with Professor Michael T. Bowser developing micro-free-flow electrophoresis for bioanalytical separations and assays. His interest is in applying mass spectrometry to bioanalytical separations, and this led him to a postdoctoral position in the laboratory of John R. Yates III at the Scripps Research Institute. He is now an application scientist with the SCIEX Separations and a professional research collaborator with the Scripps Research Institute. His current work focuses on applying capillary electrophoresis separations and mass spectrometry to the analysis of proteins and post-translational modifications. And with that, I'm going to turn our presentation now over to Brian.
Thank you for the nice introduction, Stephanie. Good morning, everyone. Thank you again for joining us. Today I'm going to be talking about comprehensive analysis or characterization of a monoclonal antibody in a single analysis. And if you're joining us, you're probably well aware of the challenges of characterizing monoclonal antibodies due to their complexity, particularly those of therapeutic nature, since there's a number of different modifications that are introduced both biologically and purposefully to improve efficacy. I'll briefly highlight some of those challenges and talk about how we have current solutions using capillary electrophoresis for the characterization of these monoclonal antibodies in biopharma. And then I'll talk briefly about linking CE to mass spectrometry, particularly for the improvement of sensitivity and dynamic range in some of these analyses. This then will lead into our CESI technology, and I'll talk about the application of this in the characterization of a monoclonal antibody. And then at the end, I'll just briefly summarize what I've talked about. So as I mentioned, and as you probably are well aware, monoclonal antibodies have a large number of modifications on them that really contribute to their complexity and also to the challenges in characterizing them. These types of modifications can be critical quality attributes if they are end up leading to issues with efficacy, patient safety, or degradation of the monoclonal antibodies. And these are really just a list of potential quality attributes that can become critical if they actually contribute to any of those issues with monoclonal antibody. So to better describe these modifications, you can see some of them listed here, such as pyroglutamate formation, degradative post-translational modifications such as deamidation, oxidation, glycosylation, isomerization, and many others listed here. So these are some of the post-translational modifications we'll be talking about today. And just to briefly highlight, there's both, as I mentioned, these types of modifications that are introduced biologically, and also those that can be introduced throughout the process. So we're very interested in being able to use capillary electrophoresis to monitor these modifications throughout the process and generation of these monoclonal antibodies. So this slide reiterates a bit of that concept. Essentially, the figure on the left shows very similar modifications shown on this monoclonal antibody, and I'll point you to this reference at the bottom if you are not entirely aware of many of these modifications. But essentially, there's a number of monoclonal antibodies that have these modifications, and now with the introduction of other monoclonal antibodies such as biosimilars and biobetters, which are acting to mimic some of the therapeutic monoclonal antibodies that have been on the market and are coming off patent, and also ADCs such as antibody drug conjugates, which are adding an additional organic molecule typically to the monoclonal antibody to improve efficacy and potentially safety. So the figure on the right actually starts to illustrate some of those instances also. And really, to summarize this concept, essentially the improvement of monoclonal antibodies is actually making them more difficult to characterize. So by adding either purposeful or unintended modifications through these processes, it becomes more important to characterize them and have technologies that are have greater sensitivity and dynamic range for these analyses. So capillary electrophoresis is an analytical separation science in itself, and in the case of monoclonal antibodies, it actually has a lot of advantages. So I'll briefly describe capillary electrophoresis in general for those that may be unfamiliar with it. So the capillary electrophoresis has the basis of the separation is through differential migration of molecules in an applied electric field. So you can see in this upper figure on the right, there's an electroosmotic flow that's generated. I'll try to use this arrow here to point this out. So by applying an electric field, you actually have this electroosmotic flow generated, and this is the bulk flow in capillary electrophoresis, unlike nano-LC or other LC methods that use pressure-driven flow. And so one of the advantages of this is it gives a flat flow profile, which gives you very good efficiency in your separation. So in addition to separating things based on charge, as shown in this image, by applying this electric field, you are also getting very good efficiencies. And so this is shown in this comparison between peaks from capillary electrophoresis and, in this case, high-performance liquid chromatography separation. So capillary electrophoresis in general is a more efficient separation technique. And to go back to this upper figure, the electroosmotic flow is used to actually drive, generally it's used to drive all of the molecules through a detector, through a detection window, or out the end of the capillary in the instance of mass spectrometry with electrospray ionization. There's other advantages in terms of small injection volumes and also minimal run buffer requirements. And there's a number of different modes of capillary electrophoresis, which I'll begin to describe shortly, that gives good separation flexibility for different types of analyses. And ultimately, it's an automated quantitative technique. So I'll highlight in a couple slides how the fact that it's automated and quantitative makes it a great alternative and improvement to doing separations with gel electrophoresis. So these are the current kits that we have for this established assessment of IgG purity and heterogeneity. And these all use capillary electrophoresis. So in the first case, lower left, this is a kit that's used for doing SDS gel electrophoresis, which allows you to do purity and heterogeneity analysis of a monoclonal antibody, both in the intact and reduced forms. In the middle, there's also isoelectric focusing kit that allows you to do charge heterogeneity analysis. And on the right is a glycan analysis kit, which allows you to do glycan or carbohydrate profiling and analysis for a monoclonal antibody. and that is of the released glycans. So there's a number of kits here already that have proven quite useful in biopharma, and I would urge you to look further into these if you're having current challenges with some of the methodologies you've used. And here's some examples of the results you might expect from those type of analyses. As I mentioned, the improvement over gel electrophoresis, slab gel electrophoresis, there's an image here showing a separation of a monoclonal antibody. I believe this is a reduced form showing the heavy and light chains, and the comparison to that with capillary gel electrophoresis. So you can see you get a very similar profile, and in this case, it's automated and quantitative. Similarly, there's the CIF and also an analogous capillary zonal electrophoresis kit, which allows you to do charge heterogeneity analysis. And this works quite well for determining isoelectric points of charge isoforms of your monoclonal antibody. And finally, the glycan analysis kit, which allows you to profile the released and fluorescently labeled glycans from your monoclonal. And just to highlight, this CE has become starting to become a gold standard for these type of analyses, as illustrated in this lower box here, showing that USP actually suggests this as in revisions of methods to include these type of methodologies. Another nice advantage of using CE is that it's reliable, robust, and transportable across sites. So you can see from a number of publications here and a few that were presented at CE Pharm and pending publication, that generally all of these methods with capillary electrophoresis can be used across different continents and among different companies to characterize these monoclonal antibodies. So this will become more and more important, especially with the biosimilar and biobetter markets. So what we've learned from the P800 Plus system, which I highlighted with the kits I showed earlier, is that within the quality by design process of monoclonal antibody generation, that we found it works quite well generally in analytics, and as shown in this figure, is well integrated into about 2/3 of the process, and it's worked quite well for these characterizations as it's fed down the line. But what we're learning is that greater sensitivity and throughput is necessary earlier in these processes, and so we're excited about, as I believe I'll describe in the next slide, the capabilities of coupling capillary electrophoresis with mass spectrometry to start to address some of these challenges that are further upstream in the quality by design process. So here shows our new CESI-8000 capillary electrophoresis system for doing mass spectrometry coupled to the ABSciX 5600 triple top system. And you can see that they're coupled here in the middle through a specially designed interface that clicks into the nanospray source on a 5600 mass spectrometer. And what's really important to note about this connection is that it's at an extremely ultra-low flow rate. So these separations and electrospray ionization processes are being done in a flow rate range that's less than 30 nanoliters per minute. And so over the next couple of slides, I'll start to illustrate why this flow rate is so important. But essentially, what we're doing is we're coupling high-resolution capillary electrophoresis separations with high-resolution and high-sensitivity mass spectrometry on this 5600 triple top system. So the way this capillary electrophoresis process is being integrated with electrospray ionization and ultimately through mass spectrometry is with a porous tip. So this figure here illustrates what we're calling CESI, the integration of capillary electrophoresis with electrospray ionization into a single dynamic process within the same device. And this is important because this is fundamentally different than other methodologies for doing capillary electrophoresis with mass spectrometry. In this case, what's done is a capillary is etched to the point where the tip is porous. So you can see this porous segment of the capillary here. And what this allows us to do is make an electrical connection with a electrode. This would be essentially a standard electrospray ionization needle through a conductive liquid that's introduced in between this porous tip and the metal needle. And so as a result, when we're doing electrospray ionization, we're actually only introducing the flow from the CE separation into the mass spectrometer. And that's really why we get these ultra-low flow rates at less than 30 nanoliters per minute, because we're capable of only introducing the flow from the CE separation. And I forgot to briefly mention on that last slide, if you're more interested, you could look at the publication by Moini, which was shown. So the way this is integrated then and implemented is through a commercial interface. So that design has now been manufactured into this CSI 8000 cartridge. And some of the advantages of this are, as highlighted at the bottom of the slide, that there's really just a single capillary that's running from the inlet of the cartridge all the way to the outlet. And essentially there's the etched tip at the very tip of the end of the capillary, and at the inlet, that is the same capillary. So because of that, this single capillary design allows this to be done, these low flow rate separations to be done with no fluidic connections, and it really simplifies then the nanoflow separations with mass spectrometry. So we're not only getting the advantages of capillary electrophoresis separations, which I highlighted as being more efficient than LC separations, but we're also getting these ultra-low nanoliter per minute flow rates. Something else notable about these cartridges is that they have retractable protective housings on both the capillaries that are at the inlets of the capillaries and at the outlet. And so just again, pointing to one of these, that's one of the retractable housings, protective housings. And this other capillary, actually on the right side that I just pointed to, is what is used for introduction of the conductive liquid, which allows for connection of the electrical connection of the electrospray voltage to that porous tip, that porous segment of the capillary. Okay, so why are these ultra-low nanoliter per minute flow rates so important? And what this really comes from is there's a large body of literature that shows that going down to ultra-low nanoliter per minute flow rates is really important to improve sensitivity, reduce ion suppression, and ultimately ionize some really challenging molecules. So this last publication is a review, and it shows that a lot of these really challenging biological molecules for electrospray ionization, that this essentially nano electrospray has a lot of improvements for doing these types of separations and ionizations and analyses of these molecules. So if you just conceptually think of how electrospray works, the reason these ultra-low flow rates are so important is due to the fact that you get better ionization efficiency and reduced ion suppression through better desolvation of these molecules. So if you look at a high flow condition here, these droplets that are coming out of whatever separation you're performing, they're large and they require more energy to desolvate into ions. And so when you go to these ultra-low flow rates, the droplets are smaller, and ultimately the ions are generated more quickly and more efficiently. So by going to these ultra-low flow rates, we're getting, as you'll see in some of these results, really increased sensitivity and decreased ion suppression. And these results just illustrate this. So this is a infusion of angiotensin at different flow rates. And you can see that when you're at flow rates that are high, you have very low sensitivity in terms of counts per mole. But as you go to lower flow rates, you see this exponential increase, and ultimately this maximization of sensitivity at around 10 nanoliters per minute. So you can see that it's really advantageous to be below 50 nanoliters per minute and operating in this high sensitivity electrospray ionization regime. And just to put this into context with some of the other current separation technologies that are available for doing analysis of biomolecules in general, you can see that the CESI-MS is really the only one that's capable of doing these types of separations and ultra-low flow ionization processes. So even nanoflow LC-MS is still above that kind of 50 nanoliter per minute cutoff range where you start to reduce sensitivity. To continue illustrating the benefits of these ultra-low nanoflow rates, you can see a comparison of equimolar infusion of neurotensin, which is a peptide, and maltoheptose. Maltoheptose is shown on the right, which is a difficult to ionize molecule. And so by doing this at ultra-low flow rates, you can see we get equimolar, when you mix them in equimolar amounts, this group found that they get equimolar response. But when you go to high flow rates, the maltoheptose is essentially absent. So there is still some ionization, but you can see that it's surely not an equimolar amount. So you already are maybe starting to get the idea that there are surely some benefits of doing these ultra-low flow separations in electrospray through looking at some of these more challenging molecules like maltoheptose. And this is quite similar in structure to glycans, which you can imagine would have some benefits for glycopeptide analysis. So later in this presentation, you'll see the benefits of this with glycopeptide analysis. Additionally, focusing on peptides again, phosphopeptides are known to be some of the more challenging molecules to analyze. You can see that with increasing numbers of phosphorylated residues on these synthesized peptides, that the ionization efficiency goes down dramatically as you increase the number of phosphates. And this was done at 100 nanoliters or greater than 100 nanoliters per minute. But when you do the same experiment of infusion at 10 nanoliters per minute, you can see that some of these very difficult to ionize phosphorylated peptides actually have better response. And if you go to a different charge states, such as this doubly charged version of the same peptides, you can see that they actually have essentially equimolar response. So again, this illustrates the advantages of ionizing difficult to ionize molecules using these ultra-low flow rates. So there's other advantages also with just using the CSI 1000 as the infusion device, including looking at protein dimer ligand complexes. So in this case, this is a published example of mixing a beta ring protein with a P14 peptide. And you can see with these ultra-low flow rates and the appropriate buffer that you're actually able to measure each of the stoichiometries of those binding interactions. Similarly, looking at large protein complexes is another great possibility for this CSI 8000 device. You can see that the analysis of hemocyanin as different complex forms, with the largest being 18 mer of 1.3 megadaltons, and also other forms of dodecamer and a hexamer. With this analysis being performed at 10 nanoliters per minute, essentially you use only 74 nanoliters during this time of analysis, which is dramatically smaller than the equivalent 100 microliters that would normally be used in an ESI experiment. So the CSI system allows to do these types of infusion experiments where you can monitor complexes and actually ionize them without much difficulty. So you can imagine that the analysis of large molecules would be beneficial with with monoclonal antibodies. Okay, so I'll shift gears a little bit now and get specifically into the characterization of biologics, which I'm sure you guys are most interested in. In this case, we'll focus on a monoclonal antibody. I had previously highlighted some of these modifications that occur on monoclonal antibodies, and I'll show these now as potential critical quality attributes. And you can see that there's a number of these that we had listed before that are now highlighted in red, which are actually what we're capable of getting from a single CESI-MS analysis using a single digest using trypsin. So we'll talk more about these modifications and how we're able to get information about all of these from a single analysis. So as I mentioned, using CESI-MS, with one protease and one digest, we're getting 100% peptide coverage in less than an hour of analysis time. I'd already alluded to glycan analysis or glycopeptide analysis, and so what I'll show you is we're getting broad dynamic range identification of the amino acid linkage of these glycans on peptides, since we're analyzing them as glycopeptides. And additionally, since we're actually able to separate them using CE, so we get a migration pattern that's associated with the different sizes and charges of the glycans on the peptides. And using the accurate mass and the high-resolution MSMS of the mass spectrometer, that allows us to simplify the identification collectively. And we're also, as I alluded to, characterizing a number of these potential critical quality attributes that are shown here. So for the analysis, we're really not using any special tricks. We're doing a standard digestion of these monoclonal antibodies using denaturization using Waters Rapidgest and just standard reduction and alkylation. And so we reduce the monoclonal antibody and then digest it with trypsin. And then with those tryptic peptides, we just do a standard CESI-MS run. And from those results, then we're typically using protein pilot software to search for modifications. One advantage of using protein pilot is that we're capable of searching for more than 300 different post-translational modifications on these proteins without much challenge. There's also another piece of software that's been recently introduced called BioPharmaView, which is an efficient LC-MS and CESI-MS data processing for biotherapeutics. And so you can see just an example here with the search that we can get 100% coverage of both the heavy and the light chain. And what this also allows us to do is extract out all of the different peaks, the electrophoretic peaks here for these peptides from an analysis. And notably, the modified residues, which have post-translation modifications on them, are shown in red within the sequence. So this software can be used actually for both these peptide mapping experiments and intact protein analysis. And I believe there is a technical note available on the ABPsyc's website if you're interested in learning more about this software. So to demonstrate the characterization capabilities of CESI-MS, we've been mostly looking at Trastuzumab, which is a well-characterized monoclonal antibody. You're probably familiar with it. It's a humanized IgG1 that's raised against the HER2 receptor in invasive breast cancer. It has 2 heavy and light chains and also a heavy chain N-glycosylation consensus site at asparagine 300. And because it has a number of well-characterized post-translational modifications, we're able to benchmark our capabilities for looking at degradative hotspots, which are often stability indicators, and also glycovariants. So as you can see from these results here, with CESI-MS, we're capable of getting 100% coverage of both the heavy and light chain with 1 digest and 1 injection. And if you look closely, there's actually some additional information that's gained from overlapping peptide sequences. So you can start to get the idea that we're getting identification of both very small and very large peptide sequences using CESI-MS. And one of the advantages of, that's actually one of the advantages of using CSI is we do get these small identification of these small and very large peptides. So the largest one identified here is the 63 mer. And in comparison to LC separations, what's particularly unique about CE is that we're actually separating both the small and large peptides, the whole range of peptides with very good efficiency. So when you may often see small peptides come out in the dead volume of your LC separation, or some of these larger peptides may have poor peak shapes or intensities with LC, they actually, both of these sizes of peptides have very good efficiencies and sensitivities with CESI-MS. Additionally, since we're capable of getting these high-quality peak shapes and also MS-MS identifications, you can see in this publication by Cahool et al. That they're capable of getting unique B and Y ion fragments, which give direct information for each amino acid. So in the variable domain of the heavy chain of trastuzumab, they're able to identify 120 out of 120 of the amino acids specifically by B and Y ions in the MS-MS spectra. And also for the light chain, they showed they were able to get almost all of them, 105 out of 107. So in this variable domain region where the sequence information is very important, CSI gives you the capabilities of getting the majority of those amino acids directly identified from the peptide sequences. Additionally, so I briefly mentioned early in the presentation biosimilars. So this slide shows the capabilities of CESI-MS for comparing trastuzumab and a biosimilar of trastuzumab. So by this comparison of these peptide sequences, you can see that lysine 217 has been converted to arginine 217 within the biosimilar. So the sensitivity and sequence coverage of CESI-MS of monoclonal antibodies is really what facilitates this analysis. And at this point, I just want to briefly thank Anis Nicholas Francois at the University of Strasbourg, France, for sharing these results with us. Okay, so in addition to getting 100% sequence coverage and very good sequence variant analysis, you can also see that we're going to focus on the capabilities of using CESI-MS for characterizing degradative PTM hotspots. And some of the known ones listed here on trastuzumab, such as pyroglutamate formation, lysine truncation, methionine oxidation, and deamidation of asparagine, are common and characterized on trastuzumab, but are also very important generally in the characterization of other monoclonal antibodies. So the first example we have here shows the pyroglutamate characterization. You can see that we're easily able to separate the modified and unmodified version of those peptides with very good peak shapes and separation efficiency, and also get very high quality MS-MS spectra for the N-terminal pyroglutamate peptide. Additionally, if you look at methionine oxidation, we're also capable of characterizing that within this single analyses from these MS-MS spectra shown, which are of high quality. And in the case where methionine oxidation or methionine oxidized peptides aren't actually separated from the unmodified peptide, we're really taking advantage of the capabilities of this CSI with the ultra-low flow separation. So you can see that even though these are of low abundance, we're having no trouble still identifying them and also quantifying them, as I'll show in the next in a few slides yet. So the fact that there's really reduced ion suppression is what's allowing us to still characterize some of the any of these peptides that remain unseparated. So here's another example from this same analysis where we're able to get C-terminal lysine heterogeneity characterization. So you can see identifying and quantifying both of these peptides with or without the C-terminal lysine. And this example shows the identification and characterization of asparagine deamidation. So there's a number of different sites and peptides which were identified on the trastuzumab that we analyzed. And so you can see that notably we're able to actually separate the unmodified from the modified peptides. And why this is important is because deamidation is a modification that can occur along many different steps of the process, including during the analysis. So the capability of separating these actually can differentiate whether deamidation occurs during some form of the electrospray process or even during mass spectrometry. So for some of these peaks where you can still see some of the, you can see a deamidated form, for instance in this case under the unmodified form, that this deamidation must have occurred during, say, the electrospray process or the mass spectrometry process instead of this peak here, which wasn't already present at the beginning of your analyses. So you can also see that there's some additional peaks that arise from asparagine deamidation. And what's been quite interesting is recently Gahul et al. Have been able to show that we're able to characterize those other peaks that arise using CESI-MS. And what those are both aspartic acid formed from asparagine deamidation and also isoaspartic acid. So that is just the racemized version of aspartic acid. So the two peaks that are shown here are actually from differentiating the asp and the isoasp. So what's notable about this is if you look at the mass spectra shown here, the top one in B shows the deamidated form, and the lower one shows the aspartic acid form. So those can both be, we can both separate them by CE, and we can also differentiate them by MS-MS. However, because the aspartic acid and isoaspartic acid are isobaric or have the same mass, they'll actually have the same exact MS-MS spectra as shown in C. And so the capabilities of separating them by the CSI gives you a way of actually analyzing them separately. And the way this group confirmed this is by spiking in different amounts of the synthesized isoaspartic acid. And so you can see with this figure that essentially they're separated, and as you increase the amount of isoaspartic acid, that peak also increases and the aspartic acid one decreases, which gives confirmation of this racemization of aspartic acid. Okay, so I'd mentioned all of these analyses are being generated from single runs of the CSI from a single digest, but we've also run these in replicates. And you can see for all of these modifications I've listed, in addition, there's also lysine glycation and tryptophan dioxidation that we've looked at, that the identifications are all very, very good in terms of mass accuracy. And also in triplicates, the relative abundances of these are all very well measured from run to run. So this gives you a great way of doing relative quantification of these modifications with very good reproducibility between runs of CESI. Okay, so now I'll talk about some of surely the most challenging peptides to analyze on a monoclonal antibody, and these are the glycopeptides. There's a number of different glycoforms that are present and known on trastuzumab, and so we've been able to demonstrate that CSI is capable of characterizing these very difficult to analyze glycopeptides with very good coverage and dynamic range. So here's an example of characterizing, and again, this is all from the exact same run, just from a single triplet digest and a single analysis. We're capable of getting these separations and identifications of these glycopeptides. So you can see in the top left figure, these are the afucosylated forms of the glycopeptides that were identified. The lower trace here on the left is the different fucosylated forms, and the trace on the right shows some of the more difficult, more highly branched, less abundant. A few of these are contain sialic acid, which induces a large migration shift relative to the other glycopeptides. So notably, you can see with all of these traces, we're capable of separating these glycopeptides and getting very good sensitivity, even for some of these ones that are of very low abundance and of higher branching and potentially more difficult to identify. Similarly, because there's some peptides formed, glycopeptides formed with a larger sequence, we're also getting additional quantitative and identification information about these glycopeptides. So this slide shows the glycopeptides that were identified on the longer form of the peptide. And collectively grouping these identification and quantification results together, we're able to generate this table showing that we can identify 20 glycopeptides on trastuzumab in a single run from these same runs that we're getting these other sequence variants and degradative PTM identifications and quantifications from. And so similar to the degradative PTMs, we're also getting very good mass accuracy, again, on these glycopeptides. And you also look at the relative abundance characterization. You can see we're going somewhere from 55% down to, I believe, 0.06%, with very good relative standard deviations for each one of those in triplicate runs. So this really gives you a great capabilities for doing glycopeptide or glycan characterization on your monoclonal antibody with very good reproducibility between these runs. And again, talking about biosimilars, glycosylation characterization is going to be very important for comparing biosimilars to the innovators. And so these figures show the glycan profiling as glycopeptides of trastuzumab and a biosimilar trastuzumab B, and also cetuximab and a biosimilar of cetuximab as B. And by doing these comparisons, you can see that in both of these cases, the glycan and glycopeptide profiles are different. So again, I'd just like to briefly thank Yanis Nicolas Francois, the University of Strasbourg, France, for these great results that we were able to share with you today. So to begin to summarize, the capabilities of high sensitivity CSI-MS are that we're able to get purity analysis through these 100% peptide mapping coverages, again from a single run and a single tryptic digest. We're able to assess purity and stability, which are indicated by a number of these degradative PTMs that we discussed, and also this excellent glycopeptide coverage and characterization. And ultimately, because you're able to do this on a single analysis, you're getting all the information you need in a single separation with complete coverage, and many of these either potential or actually critical quality attributes, which ultimately saves you time. So what we're really hoping is that this will not only fit into some of the existing places that CE is used in biopharma, but that it will also work further up into the development pipeline where you may be limited by the amount of sample you have, or dynamic range, or sensitivity. Okay, so just to highlight again the benefits in general of CESI-MS, we're getting this reduced ion suppression, which really gives us the capabilities to get increased sensitivity for some of these really challenging molecules to analyze. Because we're using capillary electrophoresis, we also get this very high efficiency separation that's just inherent to CE. Also, I haven't really highlighted this too much, but because we're using the CE for these separations, which typically take 45 minutes, with a small equilibration time before each run, we're actually getting decreased injection time for these analyses. So a single analysis of this takes about an hour. Similarly, with the equilibration period at the front of the CE separation before each run, we're essentially getting no carryover between these analyses. And much of this comes from the fact that there's no solid support within the capillary. It's just an open tube. So anything you inject out of the capillary comes off, and that's also part of the reason we're getting these great coverages of all the way from small up to very large peptides from these monoclonal antibodies. Additionally, we're getting broad analyte coverage. So I showed you examples of the superior peptide mapping coverage all the way up to non-covalent complex characterization. So there's the potential for a lot of different, for using this system for a lot of different types of analyses you may be interested in. And ultimately, we're also reducing some of the artifacts because we have the electrospray ionization voltage much lower than what's typically used. We're often using around 1 kilovolt for these ESI processes, and also we're isolating the electrospray ionization voltage from any of the analytes that you're interested in analyzing through the manufactured tip that we have. So just briefly, I'll show you again. Here's the CSI-1000. One notable thing I didn't mention is this is included with a cart that can automatically raise and lower the system to match your mass spectrometer height. And again, here's an example of the cartridge, which just clicks into the CESI-1000, and then also clicks into the adapter that's designed for your mass spectrometer. Speaking of clicking it into a mass spectrometer, here are images that show the integration of this with the ABSciX TripleTop 5600 system, which I had shown briefly before. So we're capable of connecting it to the 5600, and also there is an adapter available for the Thermo Scientific cubed active mass spectrometer. So a number of the results I mentioned today were in publications, which I would point you to look at if you're more interested in looking at any of these results. But we also have 2 CSI-MS technical notes, which are available on our website, www.scix.com. So I would invite you to look at these more closely if you're interested in learning more about the capabilities of CSI-MS for doing these peptide mapping experiments to characterize all these different modifications in a single run with a single digest. So in summary, CE is a robust and reliable technique that's well integrated into biopharma development and control processes. And so we're really excited about the capabilities of CESI and how that might start fitting into these different processes, with CE already demonstrating many useful capabilities. As I mentioned before, CESI is the integration of capillary electrophoresis with electrospray ionization, serving to reduce, eliminate ion suppression and increase ion efficiency. So the capabilities of these ultra-low flow rates are really improving the analysis of a lot of these molecules we're starting to look at. And from a single run of a therapeutic monoclonal antibody digest, as I had shown, CESI can provide a peptide map with 100% coverage, detection of critical PTMs such as degradation hotspots, identification of glycovariants with ultra-high sensitivity and that broad dynamic range, as I showed with identifying 20 of the glycopeptides on Trastuzumab. And now, courtesy of Yanis Nicolas Francois, we're really excited about the capabilities of CSI for comparing these innovator monoclonal antibodies with biosimilar monoclonal antibodies. And hopefully what you've gathered from this presentation is really that flow rate matters. So in addition to being able to do these separations with CE, the fact that we're able to do these analyses at these ultra-low nanoliter per minute flow rates, you really start to see some of the molecules that are very difficult to identify with some of these traditional techniques. So hopefully you'll find that this will be useful for some of the molecules that you'll be interested, that you're currently interested in looking at and will be interested in looking at in the future. Okay, so thank you again. At this point, I will pass this over to Stephanie, who will start the Q&A session for us.
Hey everyone, this is Stephanie, and I'd like to start by thanking Brian for that informative presentation. Just a quick reminder before we get started going through your questions, you can submit your questions by typing them in the Q&A box, which is found by clicking the green Q&A button in the lower left presentation window. And we're going to try to get to as many questions as we can today. And so with that, Brian, our first question comes from Scott Wagner at BioVest International Incorporated, and he wants to know, can a client send you samples for analysis to see what kind of data we can generate? They have an autologous ADC product. And with that, I'll switch back to Brian.
Thank you. That's a good question. We do have an active demo sample analysis program. I would ask that if you are interested in submitting your samples, I guess I'm unsure of the capabilities of you contacting us, but we can surely send you an email. There's also a place on the website, I believe, where you can send us, I guess, ping us and send us a little note more with more of the details of your analysis. But we're very interested in demonstrating the capabilities with this, particularly on customer samples, which may be different than some of the things that we work with thus far. So I'll pass that back to Stephanie then.
Well, thanks for answering that. Our next question comes from Gary Lee at Emergent Biosolutions, and he wants to know, do you need to do a desalt step before digestion? If yes, how do you do that? And back to you, Brian.
Thanks. That's another good question. Typically, in the analyses that I've performed and other applications scientists have performed, we're not doing any desalting steps for these peptide mapping experiments. We do have a standard protocol that could be followed, as I mentioned, using RapidGest and some of the standard reducing and alkylating agents.
Oh, thanks. Our next question comes from Mike Malloy from GSK, and he wants to know, can you identify the glycan peptides and get a glycan profile from the experiment? Also, how does the data compare to released glycan data?
Thank you, Mike. So I may have missed part of that question, but I believe the first part was whether we can get these profiles. And so I guess just to briefly try to get back to one of these slides here. This is the case where we're comparing, a biosimilar to an innovator of Trastuzumab or Cetuximab, and you can see that we can get these profiles plotted like this, or as I showed in another slide, if I can find this quick, this table of glycopeptides that essentially give you that profile. So you can see the relative abundance of these in triplicate that show the profile then of these glycans measured as glycopeptides. And I have seen some comparison of these directly to our released N-glycan, CELIF, using capillary electrophoresis, and they indeed look quite close. I guess if you'd like to see any more of that data, we could surely get in contact with you and discuss that further. So I'll pass this back to Stephanie.
Thanks. Our next question comes from Roxana Jayo at the University of British Columbia, and she would like to know, how were the structures of the glycan portion of the glycopeptides identified, and was this performed using MS/MS analysis?
Thank you, Roxana, for the question. Yes, so I guess I didn't show that in any of these slides, but we're using just MS/MS identification of the glycopeptides. And what we're seeing is generally fragment ions for both the peptide and some of the important low molecular weight glycan structures, which give us that identification. So it's really coupling both of those things from the MS-MS with the high max accuracy measurement of the intact glycopeptide. So collectively, with all those high resolution, both MS and MS2 measurements, that's what allows us to get those glycopeptide identifications. And additionally, as I briefly mentioned, what's nice, hopefully the slide is showing up okay, so I'll show this one, that by extracting those MS1 ions over time in these extracted ion electrophoregrams, that we're actually able to see that there's a peak associated with each one of these glycopeptides. So by having all the MS-MS and MS information and also seeing a nicely shaped peak, that really gives us additional information that that glycopeptide is indeed true and there. So thank you, that was a good question.
Yep. Thanks. Our next question comes from Swin Suchard from Fraunhofer Gesellschaft. Can you tell me the amount of antibody you used for the sequence coverage estimation experiments? And back to you, Brian.
Thank you. That's another good question. Sorry, I actually forgot to mention that. For the majority of the experiments, we're using between 100 and 200 nanograms of the sample. So we would typically, prepare, 50 micrograms of sample and then perform a small injection of, 100 to 200 nanograms. And that's what allows us to get these 100% sequence coverages and additionally identify all of these different sequence variants and modifications. So from that single analysis of 100 to 200 nanograms, that's what we're capable of doing. Thank you for that question. I'll pass this back to Stephanie.
Okay. Again, apologies for any mispronunciations of names. Our next question comes from Desiree Haraharan from Aminocore Transplant. And they would like to know, what is the initial purity of the protein that's required for this type of analysis?
Thanks, that's a good question. Typically for these analyses, we're using fairly pure monoclonal antibodies. So in the case of Trastuzumab, since that's a marketed therapeutic, it's quite pure. But we're beginning to start to look at looking at impurities of these analyses could be beneficial for biopharma customers, so such as host cell proteins. But unfortunately, at this point, I can't think of any data off the top of my head that might better answer your question. But that's surely something we're interested in focusing on in the near future. So hopefully we can reach out to you and discuss that a little bit more. So thank you, and I'll pass this back to Stephanie.
Okay. Our next question comes from Marcia Santos from AB-SciX. How can you experimentally determine if oxidation or deamidation is in the sample or due to the ESI process? And back to you, Brian.
Thanks, Marcia. That's a good question. So as I mentioned, one of the ways we can do that is, we can try to find this slide quickly. One of the ways we can do this is actually by separating, in the case of deamidation, you can separate these. So if there's any form of deamidation that occurs under the asparagine peak, that's likely due to something with ESI or the mass spectrometer. And this, sorry, I should be pointing at these quickly. So essentially, this peak here shown if there's any deamidation directly under the unmodified peak, that would likely be due to something during the process or during the ESI process or the mass spectrometry process, whereas a peak that appears such as this one from asparagine deamidation is likely due to something prior to the analysis, since they were separated during the CE process. So in the case of methionine oxidation, since we're actually not resolving these, I think the way that you would attempt to, and you could also do this for asparagine deamidation, is do some kind of time course to show that this changes over time due to something that's occurring in solution versus something that's occurring during the analysis. So I don't know of any examples where we've tried that yet, but that's surely the strategy I would think that could be used for that. So thank you for that question. I'll pass this back.
Unfortunately, we are out of time for today. I want to thank everyone who attended for your interesting questions and participation in today's event. I'd also like to thank our sponsors, SciEx Separations, for making today's educational webcast possible.